Hello everyone, myself Maidil, student of Microbiology Department. Let's see about Chlamydia pneumoniae. General characteristics about Chlamydia pneumoniae. Gram's classification. Chlamydia pneumoniae classified as gram-negative cocci. It lacks peptidoglycan layer. Instead, it has lipopolysaccharides. Motility non-motile in nature. Capsule and endospores were absent. Size, it ranges from 0.2 to 0.6 micrometer in diameter. It belongs to the family Chlamydiaceae. Optimum temperature 35 to 37 degrees Celsius. Optimum pH 7.2 to 7.4. Habitat primarily present in the respiratory tract of humans. It is most commonly found in the epithelial cell lining. It causes respiratory infection including pneumonia and bronchitis. Chlamydia pneumoniae was first discovered by researchers in 1960s. It was initially isolated from the respiratory tract of military recruits. It is an obligate intracellular pathogen. They live inside host cells to survive. Pathogenicity of Chlamydia pneumoniae. Disease transmission. Chlamydia pneumoniae infection are primarily transmitted by direct contact with an infected person or by using a contaminated object, by inhaling infected droplets which containing elementary bodies dispersed by sneezing or coughing. These elementary bodies resist to harsh environment. Incubation period. It takes approximately 3 to 4 weeks to show symptoms. Virulence factors of Chlamydia pneumoniae. Chlamydia pneumoniae possesses several virulence factors. First, adhesins. Chlamydia pneumoniae uses adhesins to attach and invade host cells. Next, inclusion bodies. These provide a protected environment for bacterial replication. Next, Chlamydia pneumoniae use type III secretion system to inject effector proteins into the host cell and it manipulates the host cellular process for its benefit. Last, avoidance of host immune response. Chlamydia pneumoniae has developed a mechanism to avoid detection and distraction by the host immune cells. Clinical diseases caused by Chlamydia pneumoniae. Chlamydia pneumoniae mostly cause respiratory tract infection in humans like pneumonia, bronchitis, pharyngitis and sinusitis. Pneumonia. Chlamydia pneumoniae cause community-acquired pneumonia. Symptoms may include cough, fever, chest pain and difficulty in breathing. Bronchitis. Chlamydia pneumonia also cause bronchitis and leading to symptoms such as production of mucus and chest discomfort. Pharyngitis. This infection may also result in throat inflammation, cause sore throat. Sinusitis. In some cases, chlamydia pneumoniae associated with sinus infections. Laboratory diagnosis of chlamydia pneumoniae. Some common laboratory techniques include serological tests as PCR, cell culture, direct fluorescent antibody staining, and nucleic acid amplification test. Serological tests. This test used to detect the antibodies produced by the host in response to chlamydia pneumoniae infection. PCR. PCR can amplify and detect the genetic material of chlamydia pneumoniae. PCR is highly sensitive and specific. Cell culture. Chlamydia pneumoniae as an obligate intracellular parasite. It can be isolated by cell culture. Direct fluorescent antibody staining. This staining involves using fluorescent dyes to visualize Chlamydia pneumoniae in clinical sample. Nucleic acid amplification test. This test is highly sensitive and specific for detecting Chlamydia pneumoniae DNA in clinical sample. Antibiotic therapy and treatment. Macrolides and tetracyclines are the drugs of choice. The choice of antibiotic and duration of the treatment is depend upon the severity of the infection. Preventive measures for Chlamydia pneumoniae infection. Hygiene practices and avoid close contact and respiratory etiquette. Vaccination. Currently there is no vaccine to protect against this infection. Thanks for watching my video.